welcome everybody. Welcome. I'm in my coat because it's 15 degrees in the house. For everybody that's been here before, lovely to see you again. If there's any new faces, welcome to the Alan Turing Special Interest Group for Clinical AI. We are a group of clinicians at from various backgrounds and various sectors at various stages who have an interest in AI, and the main aims of this group is to be sharing, pooling and educating. So brilliant to have everybody here. With that, we have a fairly standard structure to these. Usually we have a headline, something of interest that's happened recently in the world of clinical AI. Then we have a couple of people introduce themselves, but if anyone's new today, feel free to also pop introductions in the chat or give us a shout if you want to do this for yourself next time. And then we have a guest speaker today as well, Professor Zoe Kourtzi. So without any further ado, I shall hand over to Nick for the headline.
Brilliant. Thanks so much, Sarah. Yes. Just one little thing to trail, which is that so I've been approached by a new journal called Touch Immunology to write a musculoskeletal clinical AI review, and I think this is a very good forum in which to be looking for some co-authors. So if anyone is interested in being part of the core writing team for the review, please do get in touch. I'll put my email into the chat, and I'm looking for all comers. So if your expertise are in data science particularly, or your expertise are in clinical and not even necessarily musculoskeletal, but you have an interest in that area, it'd be great to hear from you, and it would be great to have you involved. And I guess one of the other things just to say is that we've been meeting virtually up until now, but we're dying for a face-to-face, and it looks like our March date might well be a face-to-face, so and might be connected with in some way or aligned in some way with AI UK, which is the big Alan Turing Institute AI conference. So watch this space, but keep March free. So on to the headlines, and last time we talked about ChatGPT-3, which is this large language model that's been produced by OpenAI, and it seems to produce some really quite impressive output, which sounds very convincing, isn't always factually correct, but sounds very convincing. And in fact, Sandy Jackson, one of our members, also highlighted the potential utility in producing Python code from a set of very simple instructions, which is quite a useful one. But there's been another development in this area, and particularly in relation to medical large language models. So the Google Health team have produced a new model based on their PaLM model. PaLM stands for Pathway Large Language Model. And what they did was, if you can see here on the left, they produced this training and testing data set of questions called Multimed QA. And this consists of a number of different databases of questions, some of which are from the USMLEs, so the medical licensing, multiple choice examinations in the United States, and other multiple choice questions relating to elements of clinical medicine. But they also included an additional data set called the Consumer Health Search QA, which takes just over 3,000 common medical searches from the Google search, and looks to answer those questions. So they used this as their training data set and their testing data set on their large language model, which they were hoping would repurpose it for the clinical medical application, and then used various prompting techniques such as few shot, but also chain of thought prompting, and ended up with some quite impressive results. So this is a timeline starting in December 2020 with increasing performance of various different large language models that have aimed to perform well in MedQA, which is the USMLE examination, or a set of examination questions. And there is an impressive leap in performance for FLAN, which stands for fine-tuned language pathway language model, up to 67% from the previous best, which was about 50%. So it seemed to perform really well when answering those MCQ questions. But when they tried to apply it to the Google searches, which is looking for slightly more verbose and better explanations, it didn't perform nearly as well. So they went through the process here, which is circled in blue, which is instruction prompt tuning, a technique that was used by this particular team in order to fine-tune the model for that particular purpose. And we've got an example here. So the question that's put to it is, how long does it take for newborn jaundice to go away? And as you can see, the answer is not just fairly comprehensive, but also impressive and also correct. And it performed to essentially the same extent as humans. So with an over 90% accuracy, I think it was about 92% accuracy, and making mistakes in approximately 5% of cases, and humans were making mistakes in approximately 6% of cases. So that is the MedPalm model. And it's really, I mean, they put lots of caveats into the limitations and say that there's a lot of work to do, but it's certainly interesting to see a large amount of work in this area. And I think this is a space that we'll see developing over the next few years. I mean, if you look at that timeline, performance seems to improve markedly over just a 2-year period. So I think we'll start to see further developments. With that, I'll hand back over to Zoe for our 3-minute introductions.
Yeah, cool. So we have 2 lined up, although I think only one of the 2 is here, unless I'm just missing them. So we've definitely got Beatrice, and I can see her. Hi, Beatrice. Would you like to take the stage?
Thank you. It's a pleasure meeting you virtually. Let's see if we can meet face to face, maybe in March. So my name is Beatrice Panico, Beatrice for an easy pronunciation. I'm Italian by origin, and in Italy I started my career. I'm a medical doctor by training. I'm also a fully qualified neurologist, and I have a PhD in neuroscience. So I worked both in academia and as a clinical neurologist, and I worked also in research, working on animal models, so neuromuscular disorders, and then I moved into neurodegenerative disorders. Then I decided I wanted to have something different, so I moved into industry, and I started working in pharmacovigilance, first in Italy. Then I moved to Switzerland, and I started working in pharmacovigilance with international responsibility for an oral drug for multiple sclerosis. And then I got a nice job offer, and I moved to the UK, and I started working for the MHRA, where I worked for 9 years. And I worked at the MHRA in clinical development, and that's where I met AI. So I went to an interesting conference of the British Neuroscience Association, and I started playing with neural networks and machine learning. And I was invited to join the Birmingham group, and I was involved in the interesting discussion that resulted in the publication on the Spirit and Consort AI guidelines. As a result of this lovely interaction, I was invited to join the WHO group working on AI in health. And one day we might publish a white paper on the regulatory principles for people who want to use AI in health. And then I started working on AI at the MHRA level with the device colleagues. And I must admit that it was very interesting. I was involved in trying to create a first coordinated assessment of clinical trials of medical devices and medicinal products, but we stopped short of including AI use as software as medical devices and clinical trials of ATMPs. But who knows what will happen now. And then I keep on being interested a little bit in AI. I do what I can when I have time. Of course, I have a full-time job. As a full-time job now, I am a principal consultant. So I just continue giving advice to companies who want to develop medicinal products. And I keep on following AI. And for example, at the beginning of February, I will give a talk about AI tools that can be of use in pharmacovigilance of medicine. And so what can I say? I am interested in safety, in regulatory aspect of AI, and particularly I would like to continue discussing the issue that it is very important to make sure that when AI is used with medical purpose, particularly in clinical trials of medicine, there is a coordinated assessment of the benefit-risk profile so that both the device part and the medicinal product part are taken into consideration at the same time. So I will be happy to continue this discussion. And of course, since I'm a medical doctor, I'm fascinated by all the discussion regarding the ethical responsibility and medical liability when AI is used to assist and augment, at the moment, medical decisions. So thank you very much for your attention. And if you want, I can put in the chat my email. And it's been a pleasure. And thank you for all the interesting discussions.
That's an amazing wealth of experience, Beatrice. Thank you so much for that. I'd just open for a second if anyone wants to ask Beatrice any questions or comment, or if anyone else wants to jump in and do a 3-minute intro as well. Yeah, feel free. Yeah, Nick.
Beatrice, quick question. What can you just give us a quick, maybe 1-minute on what kind of AI tools are used in pharmacovigilance?
Well, one of the issues that is more common in pharmacovigilance, both in pre-marketing, I would say, and post-marketing, is the fact that we have big data. So, of course, if you start from adverse events, you can imagine how many adverse events are available to the reviewers and how important signal detection is. So, one of the first use is to use AI for signal detection purposes. And I can say it now because, of course, it's in the public domain, that when I was at the MHRA, I was involved in a project that is still ongoing at the MHRA, and it is the creation of a new system to assess the safety data coming from all sources, medical devices, medicinal product, blood tissues, and so on and so forth. And one of the things that we were trying to use AI for was to identify potentially a baseline, and then identify trends that were of interest, so that then there could be a targeted response in terms of proposal or risk mitigation strategies. And this is something, as I said, the MHRA was working on, and lots of regulatory authorities are working on, and several companies are trying to use different types of AI tools for this purpose. Another very common use is use of different language processing tools, natural language processing, and so on, for identification of, I would say, it starts with verbatim elements in the narratives of adverse events report, the individual case safety reports, plus it can be used to assist in improving the consistency when coding events using the MEDRA dictionary. And then, of course, if you are familiar with pharmacovigilance, there is the entire world of aggregate reporting. Now, in pharmacovigilance, there are two main schools of thoughts, if you want. That is the US-oriented and the European-oriented way. But no matter what, when preparing regulatory submission, it is really important to review aggregate data. And recently, the FDA has made a clear statement in this direction, and is proposing the use of a new query that will look at several aspects of adverse events, grouping them in a medical relevant manner. And they have created some query that they call algorithm-based query. And now you can understand that this is very short, or then using really AI to create a presentation of aggregate data for regulatory submission. So these are some of the aspects. And then another aspect that is very nice, and of course, I started discussing that, is another thing that is very common, is that now in clinical trials of medicine, wearables are everywhere. And that means big data again. And that has got lots of implication potentially in terms both of trial conduct and pharmacovigilance. So these are some of the areas.
Thank you so much. That was a tour de force.
Thanks so much, Beatrice. Great. So if anybody else wants to jump in, please feel free to jump in now. Otherwise, I think we'll move on to the next part. I'll hand over to Alicia.
Thanks, Sam. Yeah, thanks everyone. We'll just have to watch this space for digital data and pharmacovigilance, I think. We could do a whole session on that, Beatrice, if you were interested at some point. So I'd like to welcome Zoe Kourtzi, who's joining us, Professor Zoe Kourtzi from University of Cambridge and also the Alan Turing Institute. And I'm really pleased that she can make it this evening to talk to us a little bit about AI to do with better brain and mental health. So over to you, Zoe, and then we've got time for questions and things and a bit of a discussion afterwards as well too.
Great, thank you so much, Alicia and Nick and everybody for the invitation. Really great to be able to show you some of our work and yeah, get feedback. So let me just load the presentation. Right. Oops. Okay, great. Can you see full screen?
No, it just flipped. So we 1.4 was the right one. Yeah,
is that good?
Oh, wonderful.
Okay, perfect. Okay, great. Yeah, so yeah, I wanted to tell you a little bit about the work we've been doing. And the main question we asked is whether we can use AI to predict brain and mental health disorders early. Sorry, let me see how. I'm on different screens, so I need to find out how to advance the slides. Not on this one. Yep, there we go. Okay, great. So why do we need to predict early, especially when we focus in the space of dementia, which is the work I wanted to show you today? So we know that the clinical need here is that every 3 seconds someone's diagnosed with dementia. So it's really a grand and a global challenge. And about 50% of individuals, researchers that with dementia are undiagnosed and 35% are misdiagnosed. And the reason for that is there are lots of comorbidities that could be contributing to symptoms that look like dementia, but maybe the cause of the symptoms might not be really dementia pathology. For example, people might have cardiovascular disease or anxiety, depression, mood-related disorders, which are kind of age-related, and have the same symptoms as dementia, but not really have dementia pathology. The other challenge when we try to diagnose dementia early is that dementia really comprises of a range of disorders of different subtypes, and actually these subtypes have different causes in the brain and potentially need different treatment. And it's really important to be able to precisely identify and give a diagnosis of subtype to individuals so that we are able to take them through the right diagnostic and treatment pathway. So why would we want to diagnose early? It's a question that I get a lot, but I'm hoping to get less, mainly because people are concerned that we have no disease-modifying treatments. But this space is changing rapidly. So 2 summers ago, we saw the first FDA approval in principle for a drug that was showing changes in the brain, no changes in symptoms. That ended up not really progressing as we have liked, but as we would have liked. But end of November this year, we've seen a new drug being approved by the FDA after a phase 3 trial with changes in symptoms. So this landscape is really changing fast. And as we will hopefully be having more and better treatments, diagnostics are becoming more important, early diagnosis is becoming more important so that we can intervene at the right time and give the right treatment to the right patients. This precise patient stratification early is also really important because we know that 40% of dementias actually could be avoided if we have changes in lifestyle factors. And so early diagnosis, early detection means that through changes in lifestyle, we might never see symptoms later in life. So this is why we need to diagnose early and precisely. Now, what is the challenge we have? Currently, dementia is diagnosed based on clinical tests, cognitive screens, and also MRI scans. And at least in the UK, the NICE guidelines propose MRI as a key diagnostic measure. And what you see here is 2 structural scans, 2 structural brain scans. And I'm sure you can detect differences between 2 brains. And you could tell easily that the brain on the right is the brain that is more progressed in terms of disease. And that's true. So the brain on the left is from a healthy individual, and on the right is from a patient with Alzheimer's disease. So any of the clinicians I work with, they tell me that that's not a challenge. The challenge comes with this image in the middle. This brain, one could say, looks quite healthy. But actually the diagnosis, this comes from a patient that has mild cognitive impairment. And mild cognitive impairment is a kind of a diagnostic step before Alzheimer's. But this is actually the early stages of the disease where we could have quite a lot of this misdiagnosis. So at this early stage, typically the scores in clinical scales will start looking a bit lower. And there might be some atrophy in the brain, but it's really hard to discriminate by the naked eye whether what we have here is really dementia pathology or age-related decline. So we see a lot of this kind of 35% misdiagnosis in research. We see it at this stage. So what we have built is a neurodetection tool based on AI algorithms, and our tool works to diagnose as early as possible with the least invasive data that one could collect. So we have an end-to-end pipeline where the data comes out of the scanner. The brain scans, this is anatomical scans, they get pre-processed by our pipeline and they get kind of married up with the clinical data. This would be clinical scales. And then we run them through an algorithm that I will talk to you about a little bit. The algorithm allows us to make 2 predictions. One is whether we have a patient that will have dementia or not, and the other prediction we can make is about each patient's individual health trajectory. So whether the patient will be moving towards Alzheimer's disease fast or slowly. And what you see here is that that tool gives us a score. It's a predictive score, it's a probabilistic score that tells the clinician whether a patient is close or further away from being stable. And the higher kind of the score, the faster the patient is moving away from stable health to disease. So let me tell you a little bit about how this model works. So this prognostic model, one of its main kind of characteristics at this core is that it's multimodal. It works with multimodal data. So the model is trained on research cohort data, it's trained on ADNI data, and it's trained with biomarkers. So we work on gray matter density, on APOE4 and on amyloid, but also on cognitive tests. And that's the training of the model. The first thing that the model actually does is it extracts key signal in terms of gray matter density atrophy from the scans and combines it with the other data. Then based on this research data, the model learns the space and can separate patients that have a clinical diagnosis of MCI into two classes. One is stable patients, which within a given period, and in our case is 3 years, will not progress, so their clinical diagnosis remains stable, remains MCI, versus patients that they do progress within the same time window. So that we asked the algorithm to do a quite kind of fine discrimination. Is not whether we have healthy versus Alzheimer's patients, is actually at that MCI stage, we ask the model to make a fine discrimination between patients that will remain stable versus they will progress. And then we extract through a scalar projection method a score from this model that allow us to identify whether the patient is moving fast or slow, so to identify their own kind of brain health trajectory. So just to show you a little bit of the data, as I said, the model is trained on this research cohort data from ADNI on APOE4, amyloid, and gray matter. And you can see here, you can extract the weights of these factors, how much they weight in the decision of the model. And then through the scalar projection, we get a score from the model, and that allows us to classify the patients into stable, slowly progressive, or rapidly progressive. And what you see here is patients that have the diagnosis of Alzheimer's. The model actually says that they are between slowly and rapidly progressing. Patients that they are kind of early Alzheimer's, the model says that they are in the slowly progressive space. And what's interesting is patients that have no dementia diagnosis, the model says they are stable. And this is a good kind of validation of the model. We don't give the model these labels for this test data. This is out of sample validation. This is not a training. But actually what's really interesting is when we look at pre-symptomatic cohorts. So what you see here is cohorts where individuals have participated, but they don't have any symptoms, and therefore there are no clinical labels. But what you see is that the model says that some of these individuals will remain stable, but some will slowly or even rapidly progress towards Alzheimer's disease. So this is an interesting tool because, as I said, this is not a training data, this is test data. This comes from different cohorts, not just from ADNI, and we see that the model really generalizes and makes predictions. So in moving kind of in this space of being able to test the model and validate it, we've moved to actually clinical data, and this is real world data. It's not anymore research data. This is data actually from the Addenbrooke's Memory Clinic, and I think I saw Ankit. Ankit contributed to this project. He did a lot of the hard work, so thank you, Ankit. We now have 220 patients from the Addenbrooke's Memory Clinic, and we have a similar sample actually from memory clinics in Singapore that we are working with. And what you see here is the model again is trained on ADNI, but what you see here is how it predicts on patients. So this is again test data, and it's real data. And here you will see on the left many more kind of clinical labels, because of course the people who come to the memory clinic, they've been referred from their GP, and they could have they have symptoms, and they could have any diagnosis. So we don't have the diagnosis. Our collaborators, our clinical collaborators have the diagnosis. We just get the data, we run it through the model, and we ask the model to predict. And then we open the diagnosis. So what you see is that patients that have the clinical diagnosis of Alzheimer's, the model again says that they will progress slowly, some rapidly. But then patients that actually have a diagnosis of attentional memory symptoms or no Alzheimer's diagnosis or other cognitive disorders, the model says they remain stable. And an interesting case is the mild cognitive impairment patients, where the model says some of them will remain stable. So maybe we have some misdiagnosis here, and some of them have started already progressing. Now, another interesting thing that we've done here with data, and this is from, data from Singapore. In Singapore, we've been working with memory clinics where the patients have been followed for quite a long time. What we have here is up to 5 years. And these are survival curves. So what we have here is repeated assessments of these patients, and we have their diagnosis in these different assessments. So what you see is we extract the score from the algorithm, from our model for each one of these patients, and we classify them into stable, that's the green line, or progressive. Slowly is the orange line, rapidly is the red line. And then what we do is we check here to see what is their diagnosis. So when the algorithm said that they will remain stable, you see over 5 years their clinical diagnosis remain the same. And when the model said that they will be rapidly progressing, you can see that their clinical diagnosis changed. That's the red curve. Slowly progressing, you can see that that curve is changing much slower. This for us is really an exciting result. It's hot off the press. We pulled that through last week because we are submitting an AAC abstract on it. And it's really clinical validation, a really strong clinical validation. I'm always asked the question of, well, how do you know when you have to wait for the patients to follow them for at least 3 years before you know whether the model has actually predicted correctly? And this is what we have. This is kind of retrospective data, but again, it's clinical data. It's not just research data, and it comes with all the diversity and homogeneity really that you have in clinical data. So giving us great hope that these tools can actually be implemented in clinical practice and hopefully make a difference. So another domain, and I'll be finishing soon, another domain that we're looking at is clinical trials, because we believe that this patient stratification can make a difference when we run clinical trials and develop new drug treatments. We've been working here in collaboration with AstraZeneca, that they've opened data from a failed clinical trial to us. And what you see is if you look at the sample of this clinical trial, and you try to separate it based on APOE4, for example, you see that APOE4 as a marker will not separate data coming from gray matter and amyloid. But if you actually look at the same data and you separate it based on the model's predictions on the score we get from the model, you see that we have two different categories. The data is stratified nicely to rapidly versus slowly progressive, and it's really the interaction between gray matter atrophy and amyloid burden that allow us to separate the patients in two groups. And we have first actually evidence that this can make a difference in terms of in the drug trial, in terms of the rapidly progressive patients are the patients that respond more to the drug treatment. So again, potentially giving us hope that with new drug target development, if the stratification is more precise through AI tools, we will have better outcomes, because this stratification reduces the heterogeneity that we have within both the drug group and the placebo group that effectively kills the statistics. So just to briefly summarize, the tool we have has really the potential to change the clinical pathway. We can look at this kind of patient classification as being represented by a traffic light system. We have patients that are stable, and these patients can go back home. They don't need to go through a really invasive and painful diagnostic pathway. These people should not really be sent for PET scans or lumbar punctures. Then we have patients that they are slowly progressive, and they could be sent back home with some care and follow-ups. And we have rapidly progressive patients who are the patients that need potentially immediate treatments and immediate more diagnostic workup, but also follow-ups. And we hope that this really has the potential to change the current really prolonged pathway where we look at up to 18 months for diagnosis, and sometimes not even a precise diagnosis at that stage. And of course, this could make a big difference for the NHS in terms of costs, but also for patients and their families that have to undergo a very long, prolonged pathway that is, of course, quite uncomfortable, but also very anxious and uncertain. So just to summarize, we've done a first health economics assessment, looking only at how tools like this can change, can make a difference in terms of hospitalization rates in this case. And we've seen that it's actually the changes in the costs by reducing hospitalization rates, because some of these patients do not need to be hospitalized because they're stable or slowly progressive. We can have a difference in saving 500 million for over 5 years. And of course, there are many more factors than hospitalization, like expensive diagnostic tests, and also social care that can be taken into account in the space of health economics assessments. So I should close and let you ask questions, and just to say that really this is not a single person's effort by far. A huge team of fantastic collaborators, both clinical and computational collaborators, and a lot of neuroscience, and a fun team to work with. Yeah, great funding, and really great to be supported by the Alan Turing Institute in many respects on this work. Thank you.
Thanks, Zoe. It's silence, but if we were in a room, we would be clapping and championing the work that you've just presented. It's just such a fantastic example, and running from all the way from research through to real world application and the potential, and even social care got a mention, which is great as well too. If people have questions or comments or points that you'd like to ask, then just drop them in the chat or put your hand up. And Nick.
Thank you so much, Zoe. That was really, really interesting. And my primary area of research is in a similarly chronic condition in the form of osteoporosis, where you can take clinical variables and use those to produce a predictive model. And so the one that we'd usually use is something called FRAX, which gives a 10-year risk of major osteoporotic fracture, and treatment decisions are made based on that. And we're looking at using imaging combined with those factors to try and improve the prediction. With your computer vision element, how did that actually happen? Were there parameters that were just derived from the images, or was the image as a whole used and features extracted from it?
Yeah, great question. Yeah, so in this case, actually, yeah, we start from the raw imaging data, and the first thing we did is actually a simple partially square regression, where we looked for features in these images that relate to cognitive decline. So these are quite highly diagnostic, and actually I believe that one of the reasons that this model is generalized is because these features are clinically relevant. And I think we could do more in, for example, more kind of deeper networks, like deep learning approaches, to ensure that they extract features that are clinically relevant. This increases in their interpretability of the models, but also I think it will increase a lot whether the model is generalized then to clinical data. So we go through this kind of first stage where we extract clinically relevant imaging features, and then we give them to the models that do the classification. We're now working with other models that are based on graph neural networks, for example, that they start with the whole image, and they kind of apply deep learning to extract features. The beauty of this is they might extract something that you didn't expect, but then it's really important to go back and interrogate what are these features that are extracted, and try and understand why potentially they contribute. So I think this is a really interesting kind of space where, if you want, AI and computer science comes together with clinical neuroscience, for example, or you know, in general, clinical approaches. This is really important to get this right for the models to be interpretable, but also for them to generalize.
And when you do that, will you segment out certain elements of the image? Or will you just take the image as a 3D whole from the MRI scans?
Yeah, so in these models, actually, we then extract values. So we say from this kind of area of the brain, we will extract the gray matter density. And that's the values then that we give to, yeah. But there are models that they will work from sort of like, based on medical imaging, they will work from the raw image directly.
Really interesting. Thank you.
Fantastic. Thanks, Nick. And a couple of other questions. Beatrice.
Thank you very much for your presentation. Of course, you might imagine that as a neurologist, this is quite close to my heart. I was just thinking, going back to pharmacovigilance and also to being proactive in terms of pharmacovigilance, is there any appetite for you to consider adding into the training data set known adverse events for different drugs, so that one day you could use the AI tool to predict if there are subsets of patients who will be more, more susceptible to one type or another of adverse events?
Yeah, that's really really interesting. Yeah. So I mean, these models can look also at kind of patient outcome based on drug treatments. In this case, they're only kind of predictive about disease trajectories rather than outcome in terms of drug treatments. But the same models, if we have data to train them to actually predict whether a patient will respond or not to a drug treatment, that would be really interesting. And then we can put in factors like this, as you say. So this is a nice thing. There are multimodal approaches, which means they learn the interactions of different factors. And some of these factors could be biological, and some others could be, coming from all sorts of data.
Thank you. I was just thinking that, for example, if you were to one day contact the Uppsala Monitoring Center, you could do it at the WHO level, because of course they are the ones who are in charge of the ultimate oversight of what's going on. So it could be very interesting.
Yeah,
thank
you. Yeah.
Lovely. Thanks, Beatrice. Hugh.
Hi, thank you. I'm a jobbing doctor, and it's good to hear an application of AI that feels a bit closer to what I do, which is look after old patients on a general medical ward. But a couple of questions, if I may. Were you saying in the clinical cohort now, in the validation cohort, it's just one scan, or are you still having to do pairs? Are you still having to do paired MRIs?
No, just one scan. So the first scan, yeah. Okay.
Second question is, what's the lead time on average between, if you like, clinically, what the hell's the word I'm after? Significant, dangerous word, but clinically significant dementia and sufficient signal from your technique? So are you talking 3 years? Are you talking 6 years? What's the kind of, have you sense of the lead time?
So you mean in terms of from when?
So if you took somebody early and ran your algorithm, and let's say it's going kind of medium pace, what's the lag time before they are likely to have sufficient clinical symptoms to influence their daily activity, medically, if you like?
So this is a really interesting question, and I'm not sure if I can answer it because we don't have enough kind of clinical data yet.
It's just you inferred at the beginning that early diagnosis of dementia would influence outcome in 40%, and I was thinking that was an ambitious claim, but early days. Okay. Can I ask one other question? I thought the AZ data was phenomenal because you've split a responder from a non-responder class. I've done a lot of work with cardiovascular drug studies, and my goodness me, the application of that is boundaryless because you have a standardized data set. So that to me is extraordinarily interesting in trying to refine targeted use of medication in the very near term. It's just an observation. So thank you.
Yeah, thank you.
Thank you, Hugh. Really good questions. And Keith.
Hi, everyone. Hi, Zoe.
Nice
to see you again. So just for everyone's context, my name's Ankit. I'm a medical doctor by background, and now I'm an associate at Hardian Health, where we advise startups, particularly in the AI space, in bringing health tech to market. But just so serendipitously, I was also a medical student. And when I was a medical student and a junior doctor, I was in Zoe's lab developing some of this work, harmonizing some of the cohorts being used for the predictive part in the first bit with Joey, whose papers were on the screen. So it's been great to see how far it's come since Joey and I have left, and yeah, it looks really good. I had a question about the health economic modeling and what you think is the bigger value driver for this kind of technology. So the budget impact model that you showed, I assume, was mostly focused on clinical impact. Do you have any inklings of how that compares to the value in a clinical trial setting for patient selection for therapeutics prior to them joining trials and recruitment and so on?
Yeah, very good question. And the answer is no, I have no clue, I'm afraid. In terms of, I think, I don't know how much data is out there, right, to be able to do this modeling. But I mean, I think that the main things with clinical trials is that they are costly, and they are very lengthy. So you could cut down quite a lot of the cost because you stratify patients, which means you recruit then the right patients rather than many patients that just increase the heterogeneity. And the other thing is you can reduce the length of the trials. So, for example, I'm sure you know from the papers, right? When we looked at this is more in the research space, we've looked at the model predicting changes in tau pathology. And when you do this and you use the model, rather than, for example, if you look only at amyloid, we see that we can increase the efficiency of clinical trials by 30%. So that can have, a really massive kind of changes in terms of the cost of the trials and their efficiency.
Yeah,
it
absolutely.
will be, it'll be really fantastic to be able to work more in this space with real clinical trial data. To be able to validate this.
Yeah, definitely. Because that was part of the research, part of the work that we did was showing that reduction of clinical trial time was, dependent on the outcome measures you use and so on. But yeah, it was just interesting to see how it affects the clinical pathway as well. And, sort of two options where these kind of technologies can be used.
Yeah, absolutely.
Lovely, thanks, Ankit, and nice nice that you're able to join as well and see. It's always nice, isn't it, whenever you can see how pieces that you work with have progressed. Fantastic questions and discussions. I'm going to drop one in before then I hand over to Nick for kind of what's coming next. And I'm just really interested, Zoe, in there are many different routes that you could take this and what you would like to kind of focus on going forward, because we're talking about there's patterns about refining diagnosis. So whenever you looked at real world data in the NHS system, kind of different clinical labels given to conditions, individual disease trajectories from stable to progressive, and the impact there on clinical management in the NHS, or even treatment and trial studies, and even being able to identify optimum treatment windows or times that, like Hugh was referring to, about when's the optimum time to try and prevent some of that progression and the implications of that. There's so many different angles and areas, all really, really important and fascinating. I'm just interested, what would be your go-to for where it's progressing next?
Yes. Well, I mean, yeah, it's really a very busy space. There's really lots of directions. So of course, we're doing much more research in terms of being able to get the algorithms to be even more sensitive and more specific. The subtype predictions are really key for us, and that's a huge space in terms of different dementia subtypes. We'll need kind of looking at different features and potentially different combinations of features, and it's a huge space in terms of improving really the precision of the diagnosis. And then we want to move even earlier, and maybe that kind of goes back to Hugh's question. So if you want, for example, diagnose when people are in their 40s, nobody's going to want to have MRI scans or PET scans, but potentially, we can build models that harness the power of the biomarkers and combine them with digital data. So, everybody will have a Fitbit or are happy to wear a Fitbit if this kind of really helps with predictions of brain and mental health. And that potentially we can be moving into this space in terms of building models that can learn from digital data, their combination with biological data, and then make precise predictions early on. And that's where I think we could see lifestyle factors playing a huge role. And the other space is really for us, we are working in the space of kind of discriminating between dementia and anxiety-related disorders, which we know in kind of are at that border of misdiagnosis. And so that has taken our work also in looking beyond dementia and other mental health disorders. And again, the early prediction there is really important. And of course, you have a huge developmental angle into this work. But it's interesting, the challenges, the questions and the challenges are very similar. And I think that's really the interesting thing. Once you're in this space, you realize that really kind of clinical AI can be very easily diversified and then applied to many different areas. Like, you know, for a lot of our work, I found parallels with oncologists that have the same problems in terms of looking at images, like we look at images and then very similar solutions apply. So I think once we build these models, as long as they are transparent and validated, we can then apply them in different areas. And of course, you know, optimizing the models for the data and understanding the data is key. So it takes, I think, a lot of kind of cross-disciplinary work and people to come with different perspectives. And it's great. It looks like in this group you have done it already. So that's I think that's really promising and encouraging for the future.
Fantastic, Zoe, and I hope yeah, we'll definitely get you back, or if others in your team or want to come and share, you know, some of that. There's a huge group of very different backgrounds in clinical areas, and both from we have members who are from NHS backgrounds and academia and pharma and lots of others as well. So please do use this group in that way. So a couple of points there on the chat that that you can hopefully see, but a point from Beatrice about the cost benefit to being able to reduce the duration of a trial, or actually make sure that your trial participants are the ones that are more likely to potentially benefit from an intervention. And Hugh's hunch for the future, short-term pharma, medium, is that population health medicine, or is that just my understanding of that acronym?
Management. So Bayesian targeting for stratification in communities. I think that'll be attractive to the NHS now.
Yes, fantastic. So lovely. Again, thanks ever so much, Zoe, and everyone else. Really, really nice discussion and questions as well too. And then Nick, our next meeting is the 14th, Valentine's Day, 14th of February.
I know, it's a spicy choice, isn't it? Yeah. So so the next meeting is Tuesday, the 14th of February, at 6 p.m. and we're going to be joined by Adam Stevenson from Our Future Health, which is a fascinating study and a combination of NHS initiative, research funding, some private funding that aims to recruit, I think, up to, is it 5 million participants?
I'm not sure of the exact number, but it is
across
ambitious.
the UK.
Yeah.
So that's going to take a lot of analysis, and I'm sure it'll be a hugely interesting discussion from a number of different clinical areas. So that's a date for the diary. Also, just to say that between now and then, there are two things you're going to hear about. One is our forthcoming summer school, Clinical AI Summer School, which is going to kick off in July, and we'll be looking for some people to help with that. And also our supra interest groups, which are going to be announced between now and the next meeting. And one of which, Zoe, is going to be a mental health and neuroscience super interest group. So one for you to potentially be involved in if you've got the time. So that'd be brilliant. Anyway, that's everything. Thank you so much to everyone for joining. Thank you again to Zoe. Thank you for all the questions, and see you next month.
Fantastic. Thanks, everyone. Lovely to see everyone.